Hello students, I am teacher Svetlana and in our today's lecture we will learn about the modern synthetic theory of evolution which is covered in the chapter Origin and Evolution of Life. Students, as far as the origin of life is considered, it has remained a debatable topic for intellectuals at all times. Despite of the advancements in various fields like biochemistry, astrobiology, geography, molecular biology, etc., scientists are unable to ascertain the truth, and hence various theories and hypotheses have been proposed to find out the answers to the questions related to the origin of life and the origin of Earth. Students, one such theory is the modern synthetic theory of evolution. It is the result of true synthesis of all biological disciplines. Studies pertaining to genetical, ecological, anatomical, geographical, and paleontological, etc., were pursued to explain mechanisms of evolution. Also, due importance was given to both mutations and natural selections. R. Fisher, J.N.B. Haldane, and many more are the main contributors of this modern theory of evolution. Stebbins, in his book, discussed 5 key factors such as gene mutations, mutations in the chromosome structure and number, genetic recombinations, natural selections, and reproductive isolations together contribute in the evolution of new species. All individuals of some species constitute a population. The populations occur in small groups of interbreeding populations. Such small interbreeding groups of population is referred to as a Mendelian population. So to understand the modern synthetic theory of evolution, we first need to understand what is a Mendelian population. So a population or a group of all individuals which are belonging to the same species and they have the abilities to interbreed. Such a population is called as a Mendelian population. The total genetic information encoded in some total of genes in a Mendelian population is called as a gene pool. So the gene pool refers to all the genes that will be contributed by all the individuals in a particular genetic or Mendelian population. Similarly, gene pool means the total number of genes of all the individuals in a population. The gametes produced by the individual furnish a pool of genes of next generation from which the genes will be selected. So, the population is going to contribute to a pool or say a collection of genes which will contribute to the next generation. So, all the genes that can be passed on to the next generation are all present in this pool, and so the selection will be done from this pool. The proportion of an allele in the gene pool to the total number of alleles at a given locus is called as gene frequency. Students, there are a number of factors which can affect this gene pool. The migration of population effectively can alter the gene pool. The gene pool also changes due to replacement of one generation by another in the Mendelian population. Does any change in the gene pool affects the entire population? Students, the modern synthetic theory of evolution comprises of 5 main factors that are broadly divided into 3 main concepts like genetic variation. Now, genetic variations can be caused due to various aspects. One of these aspects include mutations, then we have followed by recombinations and then migrations. The second factor discussed is natural selection, and the third one is the mechanism of isolations for explaining evolution of species. So according to the modern synthetic theories, these are some of the factors that are responsible for evolution of new species. So let's see each of this individually. Students, the first factor responsible for evolution according to the modern synthetic theory of evolution is genetic variations. Genetic variations is known as the change in gene and gene frequencies which give rise to variation. Genetic variations is caused by a number of factors, which are as follows. The first factor responsible for variation is gene mutation. Now, gene mutation is a sudden, permanent, heritable change in our genes. So it's called as mutation. Mutations can occur in gene, in the chromosome, or even in our chromosome numbers. So any changes in the chromosome structure or a change in the chromosome number can give rise to mutations. Now, mutations that occur within a single gene is called as point mutation or gene mutations. Now, what is the effect of gene mutation? It leads to a change in the phenotype of the organism, and hence it causes variation. The next factor responsible for variation is genetic recombinations. Students, we know that in sexually reproducing organisms during gamete formation, there is exchange of genetic material which occurs between the non-sister chromatids of the homologous chromosomes. Now, this is called as crossing over. So, we are all aware of the phenomenon of crossing over, which happens at the time of meiosis. It produces new genetic combinations, which also results in variations. Fertilizations between opposite mating gametes leads to various recombinations, resulting in the phenotypic variations, which causes a change in the frequencies of alleles. We know that after the fusion of the male and female gamete, we have recombinations that take place and new phenotypic variations occur. So this all also contributes to our genetic variation. Students, the next factor responsible for genetic variation is gene flow. Gene flow is the movement of gene into or out of a population. Gene movement may be in the form of migration of the organism or gametes, dispersal of pollen, or segments of DNA, that is transformation. Gene flow also alters the gene frequency, causing evolutionary changes. Now, to explain this concept of gene flow, consider an individual who lives in a tropical climate, or say in a hot region, say the deserts of Sahara, and suddenly he leaves that environment and moves to a colder climate, or say to an place which is in the Atlantic. So there is a quite changes in his climate or the place that he's living. So what does this individual do? He alters the genes of the population of the Sahara Desert, and when he moves to the new place, that is in Atlantic, he contributes new genes to the population of that place. So this movement of genes is called as gene flow. Now this also alters the gene frequencies and gives rise to evolutionary changes. The next factor responsible is genetic drift. Any random fluctuation or alteration in the allelic frequency occurring in the natural population by pure chance is called as genetic drift. For example, when the size of a population is severely reduced due to natural disasters like earthquake, floods, fires, etc., it causes elimination of a particular allele from a population. Smaller populations have greater chances of genetic drift. So basically you lose, or there is a complete alteration of the genes in a population. It also will result in changes in the gene frequency. Genetic drift is also an important factor of evolution. Now, genetic drift can also occur when, suppose, in a population you just had a handful of individuals possessing a particular genes, and if that particular population is affected, or say there was a single person having a different kind of gene, but due to sudden death that gene is lost before it got transferred to the next generation. So such phenomena are called as genetic drift. So loss of genes which result in alteration of the total gene pool. Student, the next important factor responsible for genetic variations is chromosomal aberrations. The structural morphological changes in chromosomes due to rearrangement is called as chromosomal aberrations. It changes the gene arrangement, that is the order or the sequence, that also results in variations. Now, these chromosomal aberrations may occur due to one of these factors. The first one is deletion. Deletion is loss of genes from a chromosome. Have a look at this picture here. This chromosome here, you will see a small segment of the chromosome is marked and cancelled out. So this region is suddenly deleted or eliminated from the chromosome. So can you see the size of the chromosome is reduced? So this process is called as deletion. Then we have a next phenomenon which is called as duplication. Genes are repeated or doubled in number on the chromosome. Have a look at this chromosome here, this region that is marked, and in this chromosome, and next in the same chromosome you will see that that marked region is present twice. So the chromosome, that region on the chromosome has doubled up. So this increases the size of the chromosome. So this process is called as duplication. Students, the next factor responsible for chromosomal aberrations is inversion. Now, a particular segment of the chromosome is broken and gets reattached to the same chromosome in an inverted position due to 180-degree twist. Now, in this case, there is no loss or gain of the gene complement of the chromosome. Have a look at this image here. See that this region which is marked here, it gets removed out and it inverts itself, takes a 180-degree turn and gets attached in the same place. So it just gets reversed. So this phenomenon is called as inversion. The next factor responsible for chromosomal aberrations is translocation. Now the transfer or transposition of a part of the chromosome or a set of genes to a non-homologous chromosome is called translocation. It is effected naturally by the transposons present in the cells. For this, have a look at this image here. There are two chromosomes that are involved in this case. We have chromosome number 4, chromosome number 20. Can you see the regions are marked? When translocation happens, the marked regions of the two chromosomes are exchanged. That is, they change their position, interchange their position. Can you see the blue region of chromosome 20 has come and attached to the green region of chromosome 4, and the green region of chromosome 4 has gone and attached to the blue region of chromosome 20. This process is called as translocation. We also have a process called as substitution, where again you have certain genes that just get substituted. That is, a part of 20 has come and attached here, and you have a part of 4 that gets attached in between somewhere in chromosome 20. This is called as substitutions. Now for this processes, that is deletion, duplication, inversion, this involves a single chromosome, but substitution, translocation involves two chromosomes. So all this collectively give rise to chromosomal aberrations, that is changes in the chromosomal structures, which eventually contribute towards genetic variations. Thank you students for participating in this lecture. We will meet soon again with the next topic. Till then, take care, be safe and keep learning. Thank you.